In this video, I will talk about the Sanger DNA sequencing method. Look at these three nucleotide structure. The first one is called a ribonucleotide. If cytosine is attached to the ribose sugar, it is called rCTP. It is present in RNA. The second one is a deoxyribonucleotide. At the 2' position, one OH group is replaced with an hydrogen atom. It is called deoxycytosine triphosphate or dCTP, and it is found in DNA. The third one has both 2' and 3' OH groups replaced with 2 hydrogen atoms. It is called dideoxyribonucleotide, and here it is ddCTP. In DNA, dCTP can react with the next base during DNA synthesis, while the 3' OH group of cytosine and the 5' phosphate group of the incoming nucleotide participate in the DNA chain elongation. Now let's see what will happen if ddCTP is added at this position. Since it lacks a 3' OH group, it can no longer extend the DNA chain. The chain will terminate here. Now imagine in a tube where all 4 dNTPs are added along with a small amount of ddCTP, what will happen? The polymerase will normally add nucleotides one after another, allowing the DNA chain to grow. But when it encounters a G in the template strand, it has two options. It can add dCTP and the chain will continue, or it can add ddCTP, in which case the chain will terminate. The ddCTP becomes the last nucleotide at the 3' end of this DNA chain. In Sanger sequencing, also called the chain termination or dideoxy sequencing, one tube contains the template DNA strand of your interest, all 4 dNTPs, and small amount of all 4 ddNTPs, each labeled with a different fluorescent dye. Now let's see what will happen. At the first base, 2 outcomes are possible. If ddGTP is added, the chain will terminate. If dGTP is added, the chain will continue. And at the second base, again there are 2 possibilities. If ddATP is added, the chain will terminate. Note, in this case, the DNA is 1 base longer than the previous one. If dATP is added, the chain will continue elongation. In this way, we can generate many DNA fragments that terminate at different positions. Now, if we try to separate all these DNA fragments using capillary electrophoresis, we know that the shorter fragments will migrate faster than the longer one. At each fragment passes through the laser detector, it emits a fluorescent signal. For example, when a fragment emits a red light, it indicates termination at guanosine position. Next, if we see a blue light, it means the next base is an adenosine. By reading these fluorescent signals in this order, we can construct the entire DNA sequence from the chromatogram. Now, once we have the DNA sequence, what will be the sequence of the original target DNA strand? You are right, it will be the complement, but written in the 3' to 5' direction. So the sequence is CTACCGGAA. I hope you like this content. Follow my channel for more videos, and thank you for watching this video.